Hello and welcome to March's Longevity Review, where we will take a look at a study which uses low-frequency ultrasound to reverse cellular senescence and increase mouse lifespan, as well as two studies on reducing lower back pain, the first of which uses epigenetic reprogramming, while the second, out of Canada, uses Senolytics. My name is Chris Linnell, and I am the founder and president of the Canadian Longevity Association, Canada's premier longevity nonprofit. If you'd like to find out more about what we do and how you can support us, then please check out the links below this video, where you'll also find a link to the newsletter and studies being reviewed here. And if you enjoyed this video, please give it a like and subscribe to our channel. So without further ado, let's take a look at the first study. Ultrasound has a long history of use, especially for medical imaging in pregnancy. It is also being used for improved tissue healing, scar breakdown, and to increase blood flow. The researchers in this study have also found that ultrasound can kill cancerous cells, and in light of that, they decided to take a look at what its effects would be on senescent cells. Now, a reminder that senescent cells are cells which have become dysfunctional with age, turning into a sort of zombie-like state where they secrete all these detrimental inflammatory factors which cause problems throughout the body. Removing them in animals has shown beneficial effects. Numerous methods have been developed to remove those cells, such as using drugs to kill them or training our immune system to get rid of them. The potential use of ultrasound would be yet another quiver in our senolytic armatorium. But before we dive into the paper, let's take a look at a summary of the top-line results. Starting with cells in a Petri dish, they found that 15 characteristics of senescent cells were reversed by low-frequency ultrasound at 33.2 kilohertz, which included returning to growth and cell division, since senescent cells don't grow or divide, and replicative senescence was reversed, as most cells can only replicate a number of times before they become senescent. There was greater cell movement because senescent cells don't move. Autophagy, which is a cellular recycling process, was also increased. Telomeres, which shorten when a cell divides, also got lengthened. Various senescent markers were reduced, as was the senescent-associated secretory phenotype, or SASP, which is those inflammatory molecules which senescent cells release and cause all those problems. Moving to mice, in 22-month-old mice, which is the equivalent of a human in their 60s, they found that their physical performance increased, their appearance improved, and their lifespan increased. Importantly as well, long-term treatment turned out to be safe. We'll take a look at the figure you can see here a little bit later. So let's now go into the details of the study. In the first experiment, which involved cells in culture, you can see in the experimental design at the top here that they first gave the cells a drug to induce senescence. Then the cells were incubated for 22 days to ensure that most of them had become senescent. And on day 23, the cells were subjected to cyclical low-frequency ultrasound, 1.5 seconds on, 1.5 seconds off. And during this time, the cells were observed to see whether or not they divided or moved. And you can see in these four images here on day 22, day 23, day 23 after the ultrasound, and 1 day later on day 24. The red numbers indicate cells, purple indicates cells which have entered the field of view, yellow indicates cells which have left the field of view, and green indicates cells which have divided. As you can see, before the ultrasound, there was not much movement. Here, a couple cells entered the field and one cell left on day 23 compared to day 22. However, 1 day after the ultrasound, 3 of the cells had divided, 5, 8, and 13. 1 cell had left, and 5 cells have entered the picture, indicating that there was greater growth and movement. This can also be seen in this chart here, whereby before the ultrasound was given, the cell number stayed the same. However, afterwards, it increased, going from 97 to 130. And in E here on the right, you can see that the movement also increased, which reached statistical significance as seen by the 2 stars on the top. They repeated the experiment, but this time only incubating the cells for 4 days, and once again found that the number of cells increased, this time going from 136 to 171. And the movement of the cells increased once again, as you can see by this bar graph here, with the after ultrasound bar being higher than the one prior. And the movement of the cells increased as well, as can be seen by this higher bar on the right, showing the cells after ultrasound was given. There are videos of this in the paper. However, it's kind of indistinct what's actually going on, so I'm not going to show it to you. In the second experiment, once the senescent cells had been subjected to ultrasound again, they were passaged 3 times, which means that the cells were transferred to a new culture medium every 48 hours. And looking at how much the cells grew in B here on the left, the bottom line shows that the senescent control cells did not grow at all. The rising line shows the senescent cells which were treated with ultrasound did grow. In fact, they grew more than non-senescent cells after 3 passages, which is the line across the middle here. And on the right here, it shows that the treated cells became normal sized again. Senescent cells tend to be larger, and you can see on this bar graph that the black bar represents non-senescent cells, the light gray are the senescent control cells, and the darker gray are the treated cells. Initially, both sets of senescent cells were the same size, but after 3 passages, you can see that the dark gray bar shrank considerably and is closer to that of the normal cells. D and E here shows EDU positive cells, which is a marker of proliferation. And you can see that the LFU, or the ultrasound treated cells, have considerably higher levels of this marker. F and G here simply shows that the cells were not killed by the LFU. In another experiment, which is found in the supplementary material, they looked at SASP secretions, which once again are the detrimental molecules released by senescent cells. And for the 8 molecules looked at here, they were found to be significantly reduced, as can be seen by the dark gray bars being significantly lower than the light gray bars. They also looked at telomere length in I and J here, looking at 2 different types of cells. And in the first one, in I, telomere length was significantly longer after 22 passages in the treated cells, as can be seen by this bar here. However, in the other set of cells, although the telomere length was a little bit longer, this did not reach statistical significance. They also looked at replicative senescence, whereby normally cells stop growing after a certain number of divisions. And whereas the control cells stop growing after a certain number of passages, the treated cells in blue here continued to expand. In addition, the size of the cells was smaller, as you can see in B here. And they had lower levels of beta-gal, which is a senescent marker, as you can see in C here. So far, the experiments took place in cultured cells. However, then the researchers moved on to mice, specifically 22-month-old mice, which are the human equivalent of someone in their 60s. And they treated the mice by placing them in a pool of water before subjecting them to ultrasound for 30 minutes. 5 days after treatment, some of the mice were sacrificed and their kidneys and pancreas were analyzed for senescence. According to the tissue staining and the bar graphs at the bottom, levels of beta-gal, again a marker of senescence, were significantly lower in the treated mice, as can be seen by these bars being significantly lower than the control bar on the left for both pancreas and kidney, indicating that ultrasound treatment reduced senescence in living animals and not only just in cells. But the researchers also wanted to find out how the treatment affected their physical performance. In this experiment, the mice were treated with ultrasound for 14 days, given a 2-week break, and then treated for another 14 days before they were tested. There were 5 groups of treated mice with varying levels of ultrasound. Those 5 groups were daily at standard power, daily at 30% greater power, daily at double the power, every second day, and every third day. And they were treated for 30 minutes each time. In the treadmill test on the left here, which saw mice run on a treadmill until they became exhausted, the black bars represent pre-treatment and the gray bars are after 4 weeks of treatment. The control mice are on the left-hand side here. And although the control mice did improve, this did not reach significance. However, for all 5 treated groups, their improvement did reach statistical significance. As there were only 10 mice in each group, both males and females, it only reached significance when the male and female results were combined. However, in both cases, there was an improvement nonetheless. For the inverted test on the right here, whereby the mice had to hang from a wire and they measured how long they could hold on for, only some of the treated groups saw an improvement, which were the every third day, every second day, and daily at twice the power. Now comes the fun part, which is what effect ultrasound had on lifespan. Those same 5 groups of mice were treated with ultrasound for 300 days. And here you can see the survival curves, with the black line being the sham or control mice, The red line being daily treatment at 2 times the power, the blue line daily treatment at 30% extra power, the green line daily treatment at normal power, the pink line treatment every second day, and the light blue line being treatment every third day. And as you can see with all these colored lines, the treated mice all had their survival curves pushed out to the right of the black line. Though the mice with the higher dose of ultrasound did not exceed the maximum lifespan of the control mice, as you can see by the red and blue lines ending at the bottom here close to the black line. However, for the mice given a normal dose, either daily, every second day, or every third day, the maximum lifespan was pushed out far to the right. Now, the researchers don't give any numbers for the percentage increase in either median or maximum lifespan, probably due to the fact that there were so few mice in each of these groups. But they do say that for the mice given treatment every second or third day, 50% survived for 1,000 days, and that 3 of the every second day mice and 7 of the every third day mice survived for more than 3 years, which when combined together was a significant increase. Although this experiment needs to be redone with larger numbers of mice, it is intriguing to see that ultrasound treatment can potentially increase lifespan. The final data that we will look at is how much voluntary activity the mice undertook. At 29 months and 32 months, the mice were placed in cages with running wheels for 3 days, and it was measured how many times the wheels rotated. The sham mice are on the left-hand side with the white bars, and as you can see, the treated mice used their running wheels considerably more than the sham mice at 29 months, especially those dosed every second or third day or daily at double the power. At 32 months, the every third day, every second day, and double the power treatment groups used their wheels considerably more, though this was not the case with the daily and daily at 30% extra power groups here in green and blue. And second to last, let's take a look at some pictures of these mice. The mice in the top row are the controls, and on the bottom are the treated mice. These pictures were taken at 30 months. And as you might be able to see, the treated mice had much thicker fur, with the control mice starting to show bald patches on their backs. But rather than just showcase pictures, the researchers also had some video to show off, which is what we will end on. We'll look at 4 brief videos, the first 2 being control mice and the last 2 being the treated mice. Each of these videos was taken at around 30 months. So starting with the first one here, you can see it's got balding at the back. It's not moving too quickly or smoothly. Looks quite decrepit, actually. The second control mouse is slightly better, but also you can see the fur is quite thin and patchy. And here's the first of the treated mice, and these mice were treated at double the power. And as you can see, the fur is quite a lot thicker than the control animals. Also, they seem to move a lot more. And now the second treated animal. Once again, with thicker fur and quite a bit more movement than the controls. So there you have it. I think you'll agree with me that this was an incredibly interesting and intriguing study. I would never have thought that ultrasound could potentially reverse senescence and increase lifespan, not to mention improving the appearance and movement of the mice. As luck would have it, there was actually another paper out in March which showed that ultrasound could reverse senescence in chondrocytes, which are the cells which produce cartilage. And I'll provide a link to that paper below this video for you to take a look at if you wish. What's especially interesting is that this is a non-invasive procedure and is incredibly safe, not only to these mice, but also to the millions of humans who have undergone ultrasound. Obviously, the study needs to be replicated with greater numbers of mice and also tested in other animal models. But if proven to be successful in those further experiments, this should be able to move quickly into humans. And I wonder if there's a future whereby ultrasound could be installed in our homes, in our bathtubs, so that every time you take a bath, not only do you get cleaned, but you can get rejuvenated at the same time. That truly would be the fountain of youth. Now let's take a look at the first lower back pain study. Lower back pain is one of the most common symptoms of aging and is a leading cause of reduced quality of life, as it can prevent people from exercising and it can limit their daily activities. Apart from injury, it is mainly caused by the degeneration of our vertebrae, in particular the intervertebral discs, which provide cushioning for our spine. There is an increase in catabolism or breakdown of tissue, as well as a buildup of senescent cells, which leads to degeneration and pain. Although pain can perhaps be managed and there are exercises one can do, there is currently no cure. However, numerous treatments are under development, and we will be looking at two of them in this and the next snapshot. In snapshot 11, we will look at a Canadian senolytic treatment. But in this episode, it is all about the use of reprogramming. First, though, a quick recap on epigenetic reprogramming. Discovered in 2006 by Shinya Yamanaka, it allows the creation of pluripotent stem cells from any cell in our body, utilizing 4 transcription factors, better known as the Yamanaka factors. But for longevity purposes, the important part is that during the process, the cell gets rejuvenated, becoming indistinguishable from a young cell. It is therefore one of the hottest fields in longevity research, the idea that we can turn back the epigenetic clock. Recently, the process has been refined, removing one of the original Yamanaka factors, the oncogene c-MYC, which reduces the risk of tumor formation. And it is this improved version which David Sinclair used to restore vision in mice. It is also the version used in the present study, which we will now take a look at, starting with the graphical summary. The study involved rats, and they used exosomes to deliver 3 out of the 4 Yamanaka factors, OCT4, KLF4, and SOX2. They are better known as OSK, however, the authors have swapped the lettering around. So in this paper, they are known as OKS. The exosomes were taken from bone marrow stem cells, and they were transfected with cavin-2, a protein which improves the uptake of the exosomes into the cells of the animal. Once OKS had been inserted into the exosomes, they were injected into the intervertebral disc of the rats. The part of the intervertebral disc that they were injected into is called the nucleus pulposus cells, which act as shock absorbers for our spine. Taking a look at what happens inside the cells, on the left we have the aged nucleus pulposus cells, or NPCs, and a lot of them become senescent with increased DNA damage, an increase in this epigenetic marker of aging called HK20ME3. They have reduced proliferation capacity, increased catabolism compared to anabolism. So again, catabolism is the breakdown of molecules, anabolism is the building up of them. So when we increase muscle, that's a result of anabolism. And there's an increase in inflammatory markers such as TNF-A alpha and IL-1 beta, which gets released from senescent cells. Once the OKS containing exosomes gets taken up into the cells, they get rejuvenated, as shown by reduced DNA damage, a reduction in this epigenetic aging marker, which results in increased proliferation capacity, a more balanced ratio of anabolism to catabolism, and a reduction in these inflammatory molecules. Now let's take a look at the details of this study. Before moving into rats, the researchers first started with cells in culture, and in the first experiment injected aging NPC cells with this OKS plasmid, which led to a reduction in age-associated hallmarks, increased their proliferation capacity, restored their metabolic equilibrium, and reduced inflammation, thereby basically turning them young again. In more detail, looking at B, C, and D here on the left, which shows 3 markers of senescence, we can see a reduction in these markers in the treated cells. So in each of these bar graphs, the red represents young cells, blue represents aging cells, and purple represents the treated cells. And as you can see, the purple bars are similar in height to the red bars. In fact, there was no significant difference between the two, but there certainly was a significant difference between the old cells and the treated cells. E and F here show age-related stress response genes, and levels are once again much closer to that of young cells compared to the old cells. G shows a marker of double strand breaks, which is when our genomes get damaged, and the treated cells have much lower levels than the old cells. H4K20ME3 is once again an epigenetic marker of aging, and here as well the levels are lower in the treated cells. Further down here, J and L show levels of beta-gal, another marker of senescence, and once again the levels are much lower in the treated cells. In M and K, EDU is a marker of proliferation, and here levels are higher than in the old cells, much closer and in fact higher than the levels in the younger cells. N and O show levels of anabolism, and here the levels are higher than in the old cells. P and Q are levels of catabolism, and here they are lower than in the old cells, closer to that of or the same as that of the younger cells. And R and S show inflammatory molecules, and once again, they are lower than the old cells and closer to that of the young cells. So this is showing that these various hallmarks of aging have been reversed in these old cells. I'll just briefly touch upon figure 2, which simply shows that the addition of this CAVIN2 protein increased the uptake of the exosomes, which we can see in G here, where the green bar is higher than the purple bar. Next, they repeated the first experiment, but this time using exosomes treated with CAVIN2 to insert the OKS into the aging NPC cells, which once again led to a reduction in the aging-associated hallmarks, an increase in proliferation capacity, a restoration of metabolic equilibrium, and a reduction in inflammation, leading to much younger NPCs. And in these bar graphs, the green bar represents these CAVIN2 exosomes. And as you can see, the green bars are the lowest for, on the left here, a senescent marker, 2 aging stress response genes, and 2 other senescent markers. In addition, at the bottom here, levels of the proliferation marker EDU were highest in the treated cells, and levels of the inflammatory marker IL-6 were lowest. I'm not sure exactly what G represents. It didn't quite explain it in the text. Now moving into animals, but starting off with some human samples in B and C with people with mild and severe disc degeneration. C simply shows that levels of P21 and P16, markers of senescence, were elevated in these patients. D shows the study design of the first treatment given to the rats. The model used to induce degeneration was by puncturing the intervertebral disc, and after 2 weeks they degenerated and became senescent. Once they had degenerated, they were then injected with the treatment or the various controls, and then 4 or 8 weeks later they were analyzed using a scanner. Now, the interesting thing about this study was that the same rat was given various treatments. So this is sort of an image of their tail, and they were either given a sham treatment, so they weren't actually punctured. They were given the OKS exosome treatment, just exosomes on their own, just OKS on its own, and sort of a saline solution. This got around the issue of variation between animals. And in the first set of results for DHI, which stands for disc height index, so how much gap there is between the actual vertebrae, either after 4 weeks or 8 weeks, and you can see that either after 4 weeks or 8 weeks, there was no real difference between the sham and the treated sections. Whereas for the 3 controls, there was a reduction in that height. And looking at the image here on the right, you can see this, the NPCs are these sort of white blobs. So that's the sham, the intervertebral discs, which weren't punctured. The OKS exosomes seem to maintain the NPCs, whereas the other 3 treated controls, this white blob sort of disappears. Here's the sort of the cross section again, white blobs here. And this was after 4 weeks, and after 8 weeks, it sort of stayed similar or the same, with the treated animals maintaining the structure. Next, in H here on the left, showing the grayscale value, which is the amount of water in the NPCs, and lower is better, showing that there's more water inside. And once again, we can see that the treated sections have no real difference, the uninjured controls, both after 4 weeks and 8 weeks, whereas the grayscale was much higher for the other 3 controls. And on the right in I here, it shows the Furman MRI score showing levels of degeneration, with 1 being lower levels and 5 being higher levels. And we can see that the treated animals, or the treated sections, show only red or blue, indicating a score of 1 or 2 after 4 and 8 weeks, whereas the 3 controls had a kaleidoscope of other colors, indicating worse degeneration. Next, we have the histological analysis of the cross sections of the NPCs, and with the sham control on the left, the treated second to the left, and then the three other sort of controls on the right here. And just sort of eyeballing it, you can see the sort of white section. Again, this is the NPCs, is much similar in the treated sections compared to the uninjured controls, whereas the other three controls fared much worse, both after 4 weeks and 8 weeks. In B here, ACAN plays a role in the development of cartilage, and I guess the staining is much stronger in the treated animals, similar to that of the uninjured sections, compared to the other three controls, both after 4 weeks and 8 weeks, indicating, I guess, that there is greater potential for cartilage growth. In C, MMP13 degrades collagen. And here the levels seem to be lower than in the other controls, and I guess more similar to that of the uninjured sections. While in D here, P21 again is a senescent marker, and judging by the level of staining, the treated sections had lower levels of this senescent marker than the other three controls. Now comes perhaps the most important experiment to see how this treatment can affect lower back pain. This time, however, instead of the tail being used, they punctured the lower spine of the animals to induce degeneration, and then 2 weeks later injected them either with the OSK exosomes or just a saline solution. 4 weeks later, 3 different pain tests were conducted. The first using pressure on the spine, the second using a sort of a wire on the hind paw, and the last one, the Hargreaves test, is a measure of thermal pain. And before looking at the results, here in B is a cross-section of the animal's spine, and with the sham, so unpunctured animals, you can see the NPC is quite clearly visible here. Whereas for the animals just injected with a saline solution, you can see that it is quite badly degenerated. However, for the animals given the treatment, once again, the NPCs are quite visible here, looking very similar to the uninjured controls. Now let's take a look at the results of the pain tests in C, D, and E, with red being the uninjured animals, purple being the saline controls, and blue being the treated animals. And 2 weeks after they were punctured, the injured animals showed much lower levels of pain threshold, which stayed low for the animals just given saline. However, there was a significant improvement in the animals given treatment, as you can see by the rising blue line. For the other 2 measures of pain, however, there was no significant difference between any of the 3 arms. Now, the researchers think that this might be due to the fact that the puncture did not rupture the outside of the disc to cause herniation and did not stimulate the nerve root to cause pain. Now, in intervertebral disc degeneration, the nerve endings expand because of an increase in nerve growth factor, which leads to increased pain sensitivity. And this hypothesis was partially corroborated by the fact that nerve growth factor did not increase in the treated animals, as we can see here in F. In these images, the red dots represent the nerve growth factor, and the saline treated animals had a lot more of these red dots compared to the treated animals or the controls, which we can also see in the bar graph on the right-hand side, where again, this is showing nerve growth factor, and the treated animals were much closer to the uninjured control animals compared to the animals just given saline, with this large blue bar here. Lastly, the researchers also looked at the transcriptomics, and without going into the details, they found that 215 genes were upregulated and 142 genes were downregulated, and those mostly sort of involved an increase in anabolic genes and a decrease in catabolic genes, indicating that there was more growth than degeneration following treatment. And that marks the end of the study, ending with the graphical summary once again. So although it wasn't a complete slam dunk in that only 1 out of the 3 pain tests saw an improvement, however, that's probably more down to the study design. What is clear, however, is that the old and degenerated cells were rejuvenated to a certain degree, and that visually the treated NPCs had the same appearance as the uninjured ones. It's also great to see another example of cellular reprogramming in live animals, as there have been relatively few published studies to date. It's also interesting that they used exosomes for the delivery of the Yamanaka factors as opposed to viral vectors, which might provide some additional benefits. And although they were injected directly into the disc, that delivery method could be used systematically to deliver the payload throughout the body. All in all, epigenetic reprogramming shows great promise, and this is yet another example of that. Let me know what you think about this study in the comments below. And now the second lower back pain study. Lower back pain is one of the most common chronic conditions of aging, affecting more than 80% of us at some point in our lives. It is a leading cause of disability and loss of quality of life. In the previous snapshot, snapshot 10, we looked at how epigenetic reprogramming could reduce lower back pain, and you can find a link to that below this video as well. In this episode, we will see if removing senescent cells can also reduce lower back pain. As a reminder, senescent cells are cells which have become dysfunctional with age, releasing all sorts of detrimental molecules and factors, which cause problems throughout the body, and their removal can produce numerous benefits. Lower back pain is often caused by intervertebral disc degeneration, and this is accompanied by a buildup of senescent cells. Therefore, the authors wanted to see if removing those senescent cells could reverse that degeneration and, as a result, reduce lower back pain. Here is a study coming from the lab of Lisbeth Haglund of McGill University. But before we go into the details, let's take a look at a summary of the results. So the authors used two senolytics, O-vanillin, which is a main metabolite of curcumin, and RG7112, which is a synthetic molecule. And both of them successfully removed senescent cells from intervertebrate disc degeneration. O-vanillin and RG7112 affect different senescent pathways, so they work even better when combined. This resulted in reduced lower back pain in mice, a reduction in the release of SASP factors. So SASP stands for senescence-associated secretory phenotype, which is those detrimental molecules senescent cells release. There was also a lower degeneration score, and the vertebral bone quality improved. In addition, there was also a reduced expression of pain markers in the spinal cord. Altogether, this indicates that removing senescent cells was beneficial for lower back pain and could potentially provide a future treatment option for lower back pain in humans. Now let's take a look at the study in more detail, starting by briefly looking at Figure 1, which simply shows that the mouse model used in this study contained high numbers of senescent cells in their intervertebrate discs. The mouse model is called SPARK or SPARK null, which means that it doesn't contain the gene for SPARK. Now, SPARK is one of the most down-regulated genes in human intervertebrate disc degeneration. And mice which lack this gene get intervertebrate disc degeneration much sooner than normal mice. They get it after around 7 months, which is very early in a mouse's life. And you can see in B here, compared to wild type or normal mice, the SPARK mice had a higher grade of degeneration both in the upper and in the lower back, as you can see by the white bar being much higher than the gray bar here. They also had much higher levels of senescent cells in the intervertebrate disc, as you can see in B here, which was especially marked in the lower back. So NP stands for nucleus pulposus cells, which is the gelatine cushioning between the vertebrates, which act as a sort of shock absorber. And AF stands for annulus fibrosus, which is the collar around that shock absorber. In E here on the left, it simply shows that there was a high correlation between the number of senescent cells and the degeneration grade, as can be seen by this line going up by 45 degrees. There are also differences in gene regulation between the SPARK and normal mice, with a greater number of senescence-associated genes being upregulated in the SPARK mice. After confirming that there was an increased number of senescent cells in the SPARK mice, the authors moved on to seeing what the results would be by removing those senescent cells. So starting off with the study design in the top left here, at 7 months of age, when the SPARK mice were already having disc degeneration, they were given Senolytics weekly for 8 weeks, during which time they were assessed for pain and then sacrificed for analysis. Both male and female mice were used, and all the results are the combined results, as the authors found no difference between the males and females. There were 8 to 10 mice in each treatment group, and as you can see on the right here, there were a large number of treatment groups. Either O-vanillin on its own with either a high or a low dose, RG7112 on its own either with a high or a low dose, and I'll call RG7112 just RG from now on. Then they had 4 combination groups, either ovanillin and RG, both at high doses, ovanillin at a high dose and RG at a low dose, ovanillin at a low dose and RG at a high dose, and finally ovanillin and RG both at low doses. In all the graphs, they use the same color scheme to differentiate them. Gray indicates the wild type or normal mice. White indicates the spark untreated mice. Blue is the high dose ovanillin. Red is the high dose RG on its own. Green is the high dose vanillin and RG together. Yellow is the high dose of ovanillin and low dose RG. Purple is the low dose ovanillin and high dose RG. And brown is the low dose vanillin combined with low dose RG. The first assessment of pain in B and C here involved grip strength. So the mice had to hang on to a wire while their tails were pulled, which would obviously cause discomfort in the lower back, and with more pain they would let go sooner. And here you can see that over 8 weeks, the wild-type mice could hang on quite well, while the spark mice started off much worse, and the untreated spark mice, which is the black line, continued to get worse. Several of the other groups improved, as you can see by these rising lines. Looking at the area under the curve for that graph, you can see that in the first 4 weeks, there wasn't much difference between the untreated spark mice and the senolytic given mice. However, as in the graph on the left, between 4 and 8 weeks, there was a divergence, with the untreated spark mice performing worst, followed by the low combination dose of ovanillin and RG. Next best were the single high doses of ovanillin and RG, as well as the low dose ovanillin and high dose RG in the purple bar here. However, best performing was the high dose ovanillin, low dose RG, and the combined high dose group. And you can see a trend emerge both in this and in subsequent graphs, whereby the worst performing treatment group was the combined low dose ovanillin and RG, whereas the best performing group was the high dose ovanillin and RG, as can be seen by this green bar here. In the second pain assessment in D and E here, where they were suspended by their tails and it was seen how long it took before they would move around in discomfort. And once again, you have the wild type mice on the top here, and the untreated spark mice at the bottom, which continued to get worse. Once again, the best performing group at the end of 8 weeks was the combined high dose treatment group, which can also be seen in the area under the curve in E here, where after 4 weeks, there's not much differentiation between the untreated and the treated mice. However, after 8 weeks, there's a clear divergence, with once again the combined high treatment group showing the best results. Though it should be noted that after 8 weeks, it did not reach the same levels as the wild type mice. Though having said that, looking at the trend lines here, if the experiment had continued beyond 8 weeks, maybe they would have reached the same levels as the wild type mice. The final 2 pain assessments were cold sensitivity and mechanical sensitivity. For cold sensitivity in F, lower is better, as can be seen by the wild type mice at the bottom here. Once again, the untreated mice and the low dose combination group got worse over time, whereas the other treatment groups improved, with the best results seen in the high combination group and the high vanillin low RG group, which is once again shown in the bar graphs on the right, especially after 8 weeks, where, as you can see, the green and yellow bars are the lowest. In the mechanical sensitivity test in H and I, whereby a thin wire is run over the hind paw of the mice, higher is better, as can be seen by the wild type mice at the top here. And once again, the untreated and low dose combination group performed worst, while the high dose combination group performed best, as can also be seen by the bar graphs on the right here. And in case you're wondering, the stars at the top here represent statistical significance, so whether or not the results could just be down to chance or were likely because of the treatment. The greater the number of stars, the higher the statistical significance. Next, they looked at the release of those SASP factors, looking at 13 specific molecules. Now, I won't go into the details of all 13, however, a lot of them are inflammatory molecules, and lower levels are better. In this experiment, there are no wild-type mice as controls, so the results are only compared against the untreated spark mice. And as you should be able to see, treatment resulted in a decrease in the release of these 13 molecules. with the low-dose combination treatment group generally performing worst, as you can see by these brown bars, while the high-dose combination treatment group performed best, as you can see by the fact that the green bars are generally much lower than the other bars. The only exception being the release of the inflammatory marker IL-2, whereby the high-dose single O-vanillin group performed best. But it is clear that treatment resulted in a decrease in the release of these detrimental factors, especially with the combined high-dose group. Next, they checked that the senescent cells had in fact been removed. So P16 is a marker of senescent cells, and both in the nucleus pulposus and the annulus fibrosus, levels were markedly reduced compared to the untreated spark mice, with once again the combined high-dose group performing best and the low-dose combined group performing worst. This was further backed up by a reduction in the degeneration grade in E here, with the low-dose combination group showing no real reduction, however, the high-dose combination group showing the most reduction. And in C and D here, it shows simply a cross-section of the intervertebral disc, and while the untreated spark mice clearly don't have any nucleus pulposus anymore, the high-treated single or combination groups maintain their intervertebral disc, confirming the previous results visually. Next, they looked at what effects the treatment would have on the rest of the spinal cord. And once again, the spark mice had a lot more senescence compared to wild-type mice based on the senescent marker P16. However, after treatment, this marker was reduced, especially in the combined high-dose group. Next, they looked at immunoreactivity and specifically 3 molecules which are associated with pain. And once again, treatment resulted in a decrease in these 3 different molecules, with once again the combined high-dose treatment group performing best, especially for this one on the right. The low-dose combination group once again performed worst. Finally, the authors looked at the intervertebral disc and the quality of the bone, and in A on the left here, you can see the difference between a normal mouse and the spark mouse, and the normal mouse has a larger intervertebral disc, as you can see with this red dashed line, whereas the spark mouse has a much smaller one, indicating less cushioning and support, and you can see by the disc volume in B here, wild-type mice have much larger volume than the spark mice. However, this volume increased upon treatment, and here we've only got 3 of the groups, so high-dose vanillin on its own, high-dose RG on its own, or the combined high-dose vanillin and RG. And just like in the previous figures, the combined high-dose group performed best, which saw a greater increase in volume, though this did not match the levels of the wild-type mice if you sort of compare. Looks like it goes to like 7.5 there, and only rises to 6 in the high-dose treatment group. Looking at cross-sections of the vertebrae, compared to normal mice, the spark mice had much greater gaps in their bones. You can sort of see these dark patches, while this was reduced in the treated mice, especially once again the combination treatment group. Before showing you the final figures below, which look at 2 specific areas of the bone, I should just quickly mention what they are. The first one is trabecular bone, and that is the inside of the bone, so cross-section here is more spongy material. And the second one is the cortical bone, which is the hard outer shell of the bone. So starting with the trabecular bone, in G to L. First in G and J, this is showing the bone volume ratio. And first we have the wild type and spark mice. You can see the spark mice have much lower volume of the spongy bone material. Whereas after treatment, this volume improved, especially for the combined high dose group. H and K shows the density. And once again, the spark mice have much lower density than the wild type mice. But this again gets improved after treatment. And I and L shows trabecular thickness, with the spark mice having lower thickness than the wild type mice. However, yet again, this got improved following treatment. Ending with the cortical bone in M to P, once again, the volume was lower in the spark mice compared to the wild type mice, which got improved in O here, as you can see by the higher bars. But this time, the higher RG single dose group performed best. Finally, in N and P here, we have MMI, which stands for moment of inertia, which is a proxy for the strength of the bone, or how it can sort of resist being bent. And once again, the spark mice had much lower strength, whereas after treatment, it improved, with once again the high dose single RG group performing best. So there we have it. Senolytic treatment clearly improved lower back pain in this mouse model and could provide a basis for treatment in humans. Importantly as well, both of these drugs appear to be safe, and I really hope that this gets tested in a human clinical trial. With this, or the epigenetic reprogramming we saw in snapshot 10, there could finally be a cure for lower back pain. Of course, what would be really interesting is to see what would happen if both of those treatments were combined. Hopefully some researchers out there will try just such an experiment. And that marks the end of another longevity review. I'd love to know what you thought about these studies, so please leave your comments below this video. And also don't forget to like it and subscribe to our channel. So until next time, goodbye.